We just finished talking about the splicing, and the next step in gene expression is going to be translation, right? So basically, the statement that describes translation is that mRNA is translated into the protein by structures that we call polyribosomes. So poly means many. So you have many ribosomes. You see those things like the little winter hats and caps, yeah. Those are ribosomes, the beige ones. The color choice on that picture is far from perfect, to say the least. Yes. Yeah, we talk about prokaryotic, and I will make that command at the end. Prokaryotic is much simpler than eukaryotic again. Now, what I'm saying, we'll look at prokaryotic because I want you to understand the basic concept. The basic mechanism is the same in pro and eukaryotes, but bells and whistles that are associated with each process, regulatory factors and so on and so forth. In eukaryotes, it's way more complicated, okay? And like they're like EIF2 alpha, eukaryotic initiation factor 2 alpha, which plays a role. I don't remember which role does it play. You know, why should you? If you work with that stuff, then it's a different story, right? You're gonna learn it anyway. But you know what? If you must know what is the basic mechanism. I want you not to lose the forest behind the trees, right? So there's polyribosomes, but polyribosomes are present both in pro and eukaryotes. That's normal. So polyribosomes are kind of clusters of ribosomes that simultaneously translate single mRNA, right? Like imagine that you have, instead of, if I want to read the book, it's only me who can read the book. But if I want you to see what's written, I will put it under the camera and you all can read the book at the same time, right? Interestingly enough, in prokaryotes, the processes of transcription and translation are continuous. What does that mean? Look at this. This beige bubble is RNA polymerase. It transcribes the DNA, which, I don't know if you can see that those really, really tiny blue lines. I highlight them for you. Okay, those tiny blue lines, DNA. So DNA, that's not correct. DNA is getting transcribed into mRNA here. And by the time transcript kind of goes out of RNA polymerase, ribosomes immediately bind to it and start translating. You see those little bead-like structures? That's protein. Imagine a writer or a poet that writes a text, and I read it while he still writes it. Okay? Does that make sense? Like you read the novel in the magazine, and they publish several chapters. Okay, imagine that you read the first chapter, but the writer is in the process of writing second one. So you read the novel as it is being composed. Does that make sense? So ribosomes translate mRNA even before the transcription is finished. Does that make sense to you? Good. Now, something I wanted to mention. Yeah, before the transcription is finished. And this is an example right here. Again, choice of colors is very suboptimal. You see that blue line, right? When I highlight it, of course you can see that. the little RNA polymerase molecules, some reason colored beige, right here, produce the transcript, mRNA. So this picture shows you different stages of mRNA synthesis. mRNA becomes longer and longer. It's rather short here and long here, right? Ribosomes immediately jump on it and start translating. You can see ribosomes here, here, here. Ribosomes translating that mRNA. Yes?
Are they moving up, or are they just attaching at one point?
This guy's probably attaching at one point, but it's really hard to say because if you think about this RNA polymerases, speaking of RNA polymerase vaguely, remotely reminds the incomplete feast, something like that. So it pulls the DNA molecule through. Does that make sense? It doesn't actually slide. It pulls the molecule through. And RNA goes out of it. That makes sense? RNA polymerase pulls the DNA, RNA comes out. Then that RNA is being pretty much pulled through the ribosomes. And speaking of which, think about this. We mentioned, it's kind of a little digression before we move on. We discussed that RNA polymerase in prokaryotes, like bacteria, is very different from eukaryotic enzyme, which means we can selectively inhibit it, right? We can make a molecule that prevents RNA polymerase right here from making RNA. And such molecule exists, and it is called rifampin. I'm going to write down the name, just FYI. And I want to ask everybody here, ever heard of it? Rifampin. Have you? I mean, there's nothing to be embarrassed about. You have not. This is awesome. It's really great. Because it's a drug against tuberculosis. Great thing that you didn't hear about it. You never ran into it. Okay, it's really one of the most effective drugs against TB. Now, that was transcription. What about translation? Ribosomes in prokaryotes and eukaryotes are very much different. Prokaryotic ribosomes are 70S, eukaryotic are 80S. Okay, so why it's so important? We can selectively inhibit ribosomes in bacteria without affecting ribosomes in eukaryotes, and we are eukaryotes. So we can make drugs that affect protein synthesis only in bacteria, only in those microorganisms, and have minimal side effects for humans. Does that make sense to you? S refers to the unit called Svedberg. So obviously, Swedish scientist with the last name Svedberg was studying how, biological macromolecules in cells, and he developed units that describe how fast or how slow different molecules or like organelles go down to the bottom of the tube, how fast or slow they precipitate. The bigger the number, the slower they to go down. Does that make sense? So if you would compare 50S subunit and 30S subunit, 50S is bigger than 30S. Does that make sense? These units are not additive. In this case, 30 plus 50 doesn't give you 70. Because it's not only about the mass of the molecule that precipitates, but also about the shape. Does that make sense? So mitochondrial chloroplasts obviously have 70S ribosomes, and it's quite amazing. they do have their own ribosomes. They carry out the protein synthesis inside of them. They have DNA, so they produce mRNA, so they produce their own proteins. Of course, those proteins are not sufficient for them to survive independently, completely out of the cell, but they still produce certain amount of their own proteins. Structurally, ribosomes are made out of proteins and RNA. They aren't membrane bound. They're not surrounded by the membrane, so just accumulation of proteins and RNAs. And interestingly enough, RNA, as I mentioned here, is the enzyme. Ribosomal RNAs not only play a structural role, they actually facilitate protein synthesis. Now, before we move on to the genetic code, this continuous synthesis, continuous processes of transcription and translation being, I don't have the good word for it, kind of being concurrent. Good, thank you, concurrent. It happens only in prokaryotes, but not in eukaryotes. Why? Why it can happen in prokaryotes, but not in eukaryotes? There you go. What about it?
It protects the DNA.
Well, it does. Well, it does protect, but eukaryotes, do they do transcription? Do they make mRNA? Sure. Do they translate? Do they synthesize protein? Absolutely. Transcription, where does it happen? In which part of the cell? In the nucleus. Translation, cytoplasm or endoplasmic reticulum. Correct? So in order for mRNA to get transcribed, mRNA first has to be delivered from nucleus to the cytoplasm or to the endoplasmic reticulum. Yeah, you just can't do it. You have membrane in between, that's it. Okay? That's one thing. What is another reason why in prokaryotes, in bacteria, mRNA can get translated right off the bat? And in eukaryotes, there's another thing that just doesn't make it happen. It's about sequence, but not proofreading. mRNA is actually, the transcription is very error prone. There's so many errors, huh? What is the difference between mRNA in bacteria and mRNA in eukaryotes? Yes, mRNA in eukaryotes have to be spliced first. It has to be edited. Does that make sense? So you gotta make a molecule, edit it, stitch it back, send it to ER, and then make a protein. Sounds like a lot of work, and it is, okay? But you just can't make it a concurrent, several concurrent processes, right? Let's go to genetic code.
So there's the two reasons why.
Yeah, that's pretty much two reasons. The spatial separation, nucleus and endoplasmic reticulum, and necessity to edit, the splicing step, okay? So genetic code. You have 21 amino acids. One girl in Monday-Wednesday class, she kept insisting that we have 22, and I almost agreed. Actually, there is some evidence that we have 22nd amino acid, but I didn't see much of it lately. So we may have 22, but 21 for sure. We have 21, bacteria have 21 amino acids, right? They're all different, structurally different. Think of them as objects, okay? Each amino acid is the object, and each amino acid has to be encoded, has to have a word. When I think about protein, I think about the statement. Like a sentence or a text. Sentence or a text consists of what? First and foremost. The big words. First and foremost, it's words. Words consist of letters. In English, we have 26 letters, and they can make thousands of words. But we combine them in different way, right? In the language of DNA and RNA, we have nucleotides. So how many letters we have? 4, yes. Let's stick to the RNA, okay? So this is going to be A, G, C, and U, 4 letters, right? Now, this language is fairly restricted. All we need to have is 21 words to name 21 different amino acids in the sequence. Does that make sense to you? So we got to have a certain combination of letters for each amino acid. Does that make sense? And if you would look at this table, you will see that these combinations are 3-lettered. Why 3? Why not 1? Why we can't have 1-letter words? Well, how many 1-letter words? Yeah, we can make only 4. That's definitely not enough for 21 amino acids. What about 2? How many 2? More. 2-letter words, 16. How many? So 16, is that enough for 21? No. How many 3-letter words can you have? Well, you can count. I can teach you a neat trick. The answer is 64. And the neat trick, it's math. And I know everybody hates math. I even had on one of the exams at a statement, math sucks. Which I kind of sort of agree with. Huh?
They like put that down as an answer for a question.
Yeah, it was kind of a part of an answer to one of the questions on that exam. But anyway, so how do we calculate how many combinations of 4 letters we can make? How many triplet combinations? Here's the little trick. So it's a neat math trick. When you take math, you can amaze your professor. They probably won't get amazed. Do you use code locks? Lock with the dials. How many? Like 3 dials. How many numbers on each dial? 10. 0 to 9. So it's going to be the lowest, the first combination. 3 zeros. Next one. 0, 0, 1. Then 0, 0, 2. So if you go like this, how many combinations at all? No. What is the last combination? 999. So 999 plus zeros, 1,000. Make sense so far? 1,000 combinations. If you decide to figure out which combination is correct, I did it on my brother's briefcase when I was 10. It took me about 25 minutes. The combination was 548. So guess what? So 1,000 combinations, right? How many dials? How many numbers in each dial? 10 in the power of 3 is 1,000. So here, yeah, 4 in the power of 3 is 64. Because you have 3 dials and you have 4 different signs.
So you take however many letters you have and then you put it to the power of how many combinations.
Yes, so if you look at the license plates, we have 3 letters and 4 numbers. 4 numbers give us 10,000 combinations. 3 letters, I don't even want to think. Technically it's 26 in the power of 3, which is a lot. And when you multiply them, it's even more than that. So that's going to be enough for a long, long time. No, it's math. I just wanted to share. I love that trick. I mean, it looks like really, really complicated, but when you break it down, it's rather simple. So we have 64 different combinations, and it turns out we use all of them, right? And if you would look at the table, you'll notice that some amino acids have several codons, like threonine, that are assigned to them. Does that make sense? They usually are different by one letter, the last. And actually, that fact that they have, let's call them synonyms, okay, decreases the chances that there is a mutation in the DNA that changes one of the letters in the codon. There's less chance that that will change the amino acid in the protein. Because the thing that we care about in the end is amino acid. If there is mutation in the DNA, but amino acid stays the same, it's okay. Does that make sense? Now, due to the fact that there are more than one codon assigned to many amino acids, the code is called redundant or degenerate. Now, imagine we have something, synonyms in English, right? We have some large, huge, vast, gigantic. We use one word more than another, right? Same goes for those codons. It turns out we have some codons that are used more frequently to encode an amino acid than others. Just interesting thing. We don't really know the evolutionary meaning of it, but some codons are preferred to others. Like out of this 4, I don't know which one is preferred, but out of this 4, 1 is going to be used more frequently than other 3. Does that make sense? Now, you have mRNA, the red stuff. You have ribosomes on them, the little beige thingies. And you have these proteins that are synthesized by ribosomes. Proteins consist of amino acids. Amino acids have to be transported to the place where ribosome sits on the mRNA to make a protein. Make sense? The structure that's responsible for that transport is called tRNA. Okay, we're gonna do a little play at the end of the class. So I need probably 4 volunteers. Nothing embarrassing, nothing like really simple thing. It's just easy to illustrate the translation, the protein synthesis. So think about who wants to be a volunteer. Now, this tRNA here, we've already seen that, carries amino acid at its tail, let's call it a tail. And here it has anti-codon loop. Anti-codon loop is complementary to a specific codon. Let's take this one, GCC. GCC codon encodes for amino acid alanine. It means that in this place in the protein should be alanine. So what is the complementary sequence to GCC? CGG. Transport RNA that has CGG and its anti-codon loop will recognize that particular codon, interact with it, and this amino acid, alanine, will be inserted in a particular place in the protein. Only matching one will get inserted. Does that make sense? Wait till we get to the plate, everything becomes so much clearer. Seriously. Okay? Now, you can imagine how many amino acids we have. Look at the little thing. That's my fault. I didn't mention this. Along with the codons that mark amino acids, we have 3 codons that mean stop. So if not 64, then 61. We have 61 different tRNAs, transport RNAs, that carry amino acids, like threonine here has 4 different tRNAs. They all carry threonine, but they all have different anticodons. Okay? Does that make sense? Now, translation. This is mind-boggling. I'm going to go through this and then we're going to execute the play. Right? This is how it starts. The small subunit of the ribosome recognizes a special sequence or structure of the mRNA. Sequence is called Shine-Dalgarno in the prokaryotes, or there's a special cap structure on the 5' end of the mRNA. This is in eukaryotes. Once small subunit and mRNA is bound, the tRNA with the first amino acid enters the complex. First amino acid is always methionine. Always. You see, AUG codon, which is also called a start codon, it encodes for methionine. And you can see amino acid here, which has anticodon matching U, A, C anticodon. To complete that complex, large subunit binds to this. So initiation complex consists of large and small subunit, mRNA, and transport RNA, the first transport RNA. So this is your initiation complex. Next step is binding of next tRNA to this site, this place in ribosome that is called A site. I usually say A stands for attachment. In the meantime, another tRNA, the previous one, that has the entire growing polypeptide chain attached to it, sits in the P site, peptide or protein. Okay? The chain, the polypeptide chain, is being transferred to the incoming amino acid. After that, these two guys shift. tRNA, which does not have any amino acids attached anymore, gets kicked out via E, exit site. And this amino acid now enters the P site. I'm accepting questions, but don't ask them yet until we get the play, because it makes much clearer, okay? When ribosome runs into the stop codon, obviously, translation stops and you get the entire protein. To make things a little bit more confusing, this UAG, is that right, UAG? Yeah, UGA stop codon, sometimes doesn't work as a stop codon. Instead, it encodes for 21st amino acid, selenocysteine. How can it happen? Imagine that you drive to the traffic light and it's red. So you're supposed to stop. There is a guy, the policeman. The light is broken and the policeman tells you, move on. You will trust the policeman, right? You will move on, right? Same goes here. If that UGA stop codon has a special RNA sequence after it, and it may be far after, that sequence, so-called SEC, insertion sequence, it tells, it's like that policeman, it tells, no, it's not a stop. You have to put selenocysteine in here. Does that make sense? Okay. Now, before we move on to the play, it is very energy consuming process. And the process is pretty fast. So each amino acid is inserted in E. coli in 500th of a second. So 100 amino acid protein will be synthesized in 5 seconds. Now imagine how many proteins are synthesized all at the same time. Probably hundreds. So cell can produce, especially a bacterial cell, can produce a huge amount of proteins in a fairly short period of time if it has enough resources. Okay? I can't record